This Week in Virology, the podcast about viruses, the kind that make you sick. From MicrobeTV, this is TWiV, This Week in Virology, episode 1287, recorded on January 9th, 2026. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. You see, I got the year right. Only 9 days in, I got the year right. Joining me today from Austin, Texas, Rich Condit.
Hi, Vincent. We've got, according to the Weather Channel, 70 degrees, according to my window, overcast. It has not yet, at least in Manchek, where I live, frozen this year, which is some kind of record. And it, a lot of people think it's nice to have these warm winters. I find them depressing because they are evidence that we're screwing up the planet. Sorry to be so negative, but there you go. That's how I roll.
Hey, it's okay to be negative. The year's already started out really badly. Here we had a couple of weeks of sub-zero, 0 Celsius. Okay, so 32 Fahrenheit. Sub-zero temperatures. It was really painful. And then in the last few days, it warmed up. If 9 degrees Celsius can be warm, so that's very good. It feels a lot easier. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And it's a gray, overcast day here, rainy, 46 Fahrenheit, 8C, and 73% humidity. So it's kind of a, not an unusual winter day here. But yeah, we had that cold snap and some snow early on, and now we've got this.
And from Madison, New Jersey, Breanne Barker.
Hi, great to be here. Here it is 49 Fahrenheit, 10 C, and also very overcast. A little warm for a winter day, I would say, but otherwise just a standard old winter day.
Yesterday on Clinical Update, we had an email from someone who, was writing. He asked a question and also wrote about how much he's liked our shows. And he said, I want to quote Breanne, I've learned a lot. I learned a lot, not I've.
Yes.
I learned a lot.
Yes, that's what one of my favorite parts about doing this is I get to learn a lot
I have this
reading and discussion.
Yeah.
memory that you've only settled into I learn a lot past couple of years.
Yeah, I had something else, and then there was an episode where I felt like I really learned a lot, and so that's what I said, and I decided to stick with it.
If you like these programs where we tell you all about science, we tell you straight science right from the papers, no lies, no disinformation. We'd love your support to do them. We have a whole lot of science shows besides TWiV on all kinds of, a lot of microbial sciences, but also some evolution and neuroscience. So check them out at microbe.tv. And if you enjoy them, we'd love your support. Microbe.tv/contribute. The Rosenfeld Lab, Amy Rosenfeld, has moved to Philadelphia, the city of brotherly love. And she is recruiting a postdoc and a technician to work on viral pathogenesis and immunobiology. The lab studies the cross-reactive anti-enterovirus immune response and its role in viral pathogenesis, as well as investigates novel platforms for antigen delivery. They combine traditional and state-of-the-art methodologies to isolate cross-species anti-enterovirus antibodies and investigate the mechanisms by which they may affect pathogenesis. They're also interested in establishing immune-competent animal models for these enteroviruses for which none exist to determine if the mechanisms of pathogenesis are conserved among this genus of viruses. Knowledge gained from this research will aid in the development and evaluation of the next generation of anti-enterovirus biologic. Our email is rosenfelda, just all letters together. There's no spaces, no dashes, none of that, no periods. rosenfelda@mlhs.org.
What institution is that?
I don't know.
I looked. It's not
I
loading.
looked it up. I couldn't find it.
Yeah.
Really? It's some kind of institute.
Well, this is
Lankenau.
news
Oh,
to me. What?
It's the Mainline Health and Lankenau Institute for Medical Research. Yeah.
This is news to me. Congratulations, Amy.
Yeah.
She got
Yeah, that's
away
great.
from the FDA and the
Yeah.
crazies over there.
Yeah.
Yeah.
So,
So,
how long? She wasn't at the FDA all that long, was she?
I think 3 years, yeah.
Oh, time flies when you're old.
Maybe 2. I don't
Okay.
remember. Then
It's
they
been an
closed my lab.
eventful few
Oh,
years.
she went the summer of, I closed my lab, what was that, 22 or something like that? So 3
Okay.
years. Yeah, well, yeah, so check it out. Brand new lab.
Yeah.
They got money. So,
Great. Hearty
project.
congratulations.
That's
Good
awesome.
for her. That's great.
And if
Great, Amy.
anybody is, looking at this job and wondering what Philadelphia is like, it's great. It's a
Yeah,
a
you know,
wonderful
I need
town.
to. I have not spent much time in Philadelphia, and my understanding is it's great.
Yeah,
I would like to go visit.
yeah. It's
Yeah,
America's biggest small town is
It's a
the
nice city. I
kind of
like it
description
very
people
much.
give.
Cool.
ASV, American Society for Virology, current ASV members should have gotten an email not too long ago about upcoming deadlines, ASV 2026. Unfortunately, it's going to be in Minneapolis at the University of Minnesota, July 27th through 30th. I say unfortunately because, as you know, bad things just happened in Minneapolis. I'm sure it will be safe when we go. Who knows? Who knows?
Who knows
Maybe they
what will be
wanna
going on then? But
shut
that's
down
not a reason not
science.
to go.
We should. I'm going to go. I look forward to going. It's a lovely city, and now is a good time to think about going to the meeting. You have to renew your ASV membership, and you have to go to asv.memberclicks.net. You can renew. You can submit an abstract, apply for a travel award, get meeting details. The abstract deadline is February 2nd. Travel award deadline February 9th. Don't procrastinate. We have one article that Alan has found, which we want to mention before we get into our papers, Alan.
Yeah, so people have been talking recently about the Denmark's vaccination schedule, and the U.S. The folks who've been put in charge of the U.S. system who have suddenly stripped down our vaccination schedule, cited Denmark as their inspiration. Look, Denmark gives so many fewer vaccines than we do to kids. Isn't that great? Let's do the same. So this is an article in Science. It's an interview with, let me get this name right. Who is the guy? Jens Lundgren, who is a public health expert who is in Denmark. So it's about why does Denmark, of all places, have this pared down vaccine schedule? They don't recommend Hep B vaccine for all the kids. They, have several other differences. And the answer is because they're Denmark, basically. They have a unique situation as a country with an extremely robust healthcare system that everybody has access to, and a very healthy population. Their rates of hepatitis B, undiagnosed hepatitis B in their general population are ridiculously low. And so the cost-benefit ratio just isn't there for vaccinating everybody against a virus that basically nobody has. The logic in the U.S. is completely different because there's a lot of undiagnosed Hep B floating around and kids can be born and exposed to it, and you want to get them vaccinated right away. And by the way, the healthcare system in the United States is a tiny bit different from the one in Denmark in terms of access and efficiency and pretty much every other good characteristic. So this is a good long interview transcribed talking about why these differences exist and why you can't just have a one-size-fits-all approach for vaccination.
So are you telling me that you can have a logical, thought-out vaccine policy?
Absolutely.
Wow.
We used to, Rich.
Oh, is that right?
Yeah.
That was yesterday.
Last week we had a good
Yeah.
policy. Now it's actually last week even not because they had already taken away Hep B.
They already,
right.
yeah.
COVID from pregnant women and children and all this stuff.
Yeah, actually, if you look at most countries, they all have different vaccine recommendations because of the specific circumstances of that country. They don't just copy each other.
Yeah.
Yeah, that's a good article.
Yeah,
He's very
thanks,
calm.
Alan. That's great.
He's very calm, And, not having universal health care in the U.S. means a lot of people don't get cared for and they get sick. And a good way to prevent that is by vaccination. So
And
it's
we're
just
even
an
further
excuse.
away from universal health care now that it's been partially defunded.
Yeah. Although they did vote on it in the House, didn't they?
They
passed
did
it.
yesterday.
Has to go through the Senate, which
Yeah.
probably won't happen. But, Denmark is tiny. It's less than the
That too.
population of New York City.
Yeah.
And they listen to what you tell them to do. They say get vaccinated, they get vaccinated. The only thing, the food is not so great. I would say U.S. food is amazingly diverse, right? Because we have
Yeah.
so many people contributing. I mean, you've got everything here. And, in cities and even in smaller places, people who come from other countries love to cook. I love it. I think just here in New York, you can get anything to eat. It's amazing. And it's really well done. So you don't have that in Denmark.
Yeah,
You have
my only complaint is it's a little cold. The one time I was there, they told me it was like the hottest day of the year, and it wasn't that hot.
Not just that, but my host, when I visited, she said, it's wonderful here, but you have to get used to it turning dark at noon in the winter.
That'd be a tough
That's
one.
a little tough because I don't like when it gets dark at 5 here in the wintertime. Okay, this turned out to be an all herpes virus program today in terms of our papers.
It just erupted on the schedule.
It just erupted. I didn't plan it, but I found a cool paper, and then I found another cool one, and it turned out they were both on herpes viruses. So the first is a paper in Science Advances. The title is Tracing 2,500 Years of Human Beta-herpesvirus 6A and 6B Diversity Through Ancient DNA. Alan, you want to do the author honors?
Sure. So a whole bunch of authors. First author is Miriam Guelil. A senior author is Christina Tambets. It will be apparent in a moment why these people are from so many places. They are from University of Vienna, University of Tartu in Estonia, University College London, KU Leuven in Belgium, Uppsala University in Sweden, an independent researcher in Estonia, University of York, UK, Edward Via College of Osteopathic Medicine in Monroe, Louisiana, University of Cambridge, Wellcome Sanger Institute in Hingston, UK, University of Leicester, UK, Estonian Literary Museum in Tartu, Komi Science Center of the Ural branch of the Russian Academy of Sciences in Syktyvkar, Russia, Peter the Great Museum of Anthropology and Ethnography of Russian Academy of Sciences in St. Petersburg, University of Turin, Tallinn University in Estonia, the Archaeological Bureau in Bilzen, Belgium, and University of Padova in Padova, Italy.
My goodness.
Yeah.
A lot of people. So,
Cool.
your herpes viruses, you're used to herpes simplex, cytomegalovirus, Epstein-Barr virus. But after a while, they got tired of giving them names and they just gave them numbers. And here we have human herpes viruses. They're beta herpes viruses, 6A and 6B. So HHV, human herpes virus, 6A and 6B. They're, of course, herpes viruses, large double-stranded RNA-containing envelope viruses, restricted to humans. They were first found in the 1980s. Okay, so this is important here in patients with lymphoproliferative disorders. HHV, so A and B are distinct, as you'll see in a moment. B infects about 90% of all children by 2 years. Sounds like a herpes virus? Yeah. And it's best known as causing roseola infantum, or sixth disease. It's a rash disease of children, and fever. It causes febrile seizures.
Which comes right after fifth disease caused by parvovirus B19, right?
And you want to know what the other four are?
Yeah.
Yeah.
Well,
Go for it.
one's got to, I don't know if I'm right. One's got to be measles.
Measles.
One's got to be
Mumps?
mumps.
Maybe?
Does it cause a rash?
Oh, rash. Oh, it's rash. Yeah, right.
Row rash disease.
One's got
I
to be
think
chickenpox.
Chickenpox is the other one. Maybe there's a bacteria in there. Brianne, look it up.
Brianne's on it.
Yeah, Brienne's on it.
I used to know this. I used to be able to stand in front of my class and.
Okay, first disease is measles.
Okay.
Second disease is scarlet fever.
Ah,
There you go.
Ooh.
Third disease is rubella.
okay.
Fourth disease looks like there's some questions about what it is. So either Philitow-Dukes disease, staphylococcal scalded skin syndrome, or Ritter's disease. And it's either staph or this random website that I found says some say this disease does not exist.
Wonderful.
And then there's 5th and 6th.
Okay.
5th is a parvo.
Yep, 5th is
Parvovirus.
parvo.
And alpha, beta, gamma herpes viruses. Characterized largely by tissue tropism. Is that fair?
I would say it's probably genome sequences, right?
Yeah. Okay. But these are these are beta herpes viruses.
Actually, it says here they're classified by replication speed, host range, and latency site.
Okay.
So alpha is simplex and VZV, fast cycles
Neurotropic.
and latency in neurons. Beta, CMV, HHV67, slow cycles and latency in secretory immune cells.
Right.
And gammas are EBV and HHV8, infecting lymphoid cells and often linked to cancer. So
Okay.
HHV8 is Merkel's, right?
Right.
Can I just interrupt for a second here? There's white noise that just started recently. This
Yeah, I hear it. So let's see. Let's go
I'm
one
going to
by
mute
one.
mine. I'm going to mute mine.
Okay,
Was it me?
yours yeah,
Okay.
it wasn't you.
All right, let me mute mine.
Yeah, it's Brienne.
It's you.
It's your air handler, right?
Perhaps.
Okay.
Yeah, it's okay. David Renato will take it out.
Okay.
Probably
Now we know
cost
whose
us-
track it's on. He can filter
It'll
that.
cost $50 extra.
Sorry, David.
So he always used to say, Dixon just cost you $150 with my extra time, taking out his breathing. That's okay, Dixon. No problem.
It's okay. Dixon's right here. He's listening.
Yes.
I was cleaning out the incubator the other day, and I found an iPad with his name inscribed in the back.
Oh, man.
Dixon Bay Palmier Vertical Farm. So I'm
Oh.
going to keep it for a while.
Yeah.
Absolutely.
Even though it doesn't work, right? So, okay, back to HHV. So HHV6B causes this 6th disease, also asymptomatic infections. And others, they've also been linked, the 6 have been linked with other conditions, a wide range, transplantation associated encephalitis, graft versus host disease, female infertility, myocarditis, multiple sclerosis, preeclampsia. And they're not really sure if the viruses cause
Yeah,
that
a lot
or-
of this is you've got a virus that's in 90% of the population from infancy, and it shows up everywhere. I mean, it's like a Kevin Bacon type of thing. It's linked to this, it's linked to that. Yeah, right.
Right. And I think that what's really interesting to think about with that is that there are increasing pieces of evidence that show relationships between some other herpes viruses and some other disease states that have come out in the past few years that have in some cases involved huge epidemiological studies. And the other paper that we'll talk about today kind of relates to that. But those are things that have come out pretty recently. But as Vincent mentioned, these viruses, HHV6A and 6B, were described in the 80s. And so we don't have those massive studies and the amount of data on them that we do with some of the other herpes viruses that we're only just now able to start to link to some of these diseases.
Okay, there's a little more on 6A and B. They're very different, epidemiologically, immunologically distinct. 6A is thought to be older, phylogenetically older than 6B. It's less prevalent in human populations, and infections occur mostly in adulthood compared to age 6B, 6th disease. But they cause latency like other herpes viruses. And, the mechanisms of latency are quite different among the herpes viruses. Herpes simplex is latent in nerve cells, EBV in B cells, and so forth. This one is different from all of them because in a certain fraction of people, the viruses integrate into our DNA in the telomeres at the ends of the chromosomes. So that's really interesting. And then it's thought that it can be latent, integrated, so the genome is quiet, and then it's periodically reactivated. It makes more virus, and that's how it's spread. It spreads to another host. Now, usually this integration occurs in somatic cells. All right, so that's the typical integration. What's really rare is integration into the germline. It's about 0.4 to 1% of the population gets this integration in the germline. So obviously they can spread to their offspring, right? Vertical transmission.
I thought this was so cool.
Well,
Right,
and
so you-
is that 0.4 to 1% who actually get this infection or who have it in their germline, so
Germline.
perhaps their parent
Germline.
could have gotten it.
Or their
Right.
distant ancestor.
Yes.
Or
Yes.
that's actually one of the
Yeah.
questions we're going to address. How long ago did this go in, and is it still going in the genome?
Yeah,
So,
how commonly does this integrate into the germline?
right.
And yeah,
So it integrates in most people somewhere in the telomere, but in the 0.4 to 1% of people, it's in the germline, and so that can influence reactivation.
And I just want to make it clear that the integration itself, regardless of where it is unique to these guys
Yeah,
as well.
that's right.
The others maintain latency by in an episomal configuration. That is, they're not actually integrated. The DNA just hangs out in the nucleus of whatever the target cell is for latency. So not only is the telomere integration unique, but integration in general is unique to these guys.
So
now
the
there
ways,
is it as far as human herpes viruses go. There's I think they mentioned Marek's disease does this
Yeah.
in chickens.
chickens, yeah, there's an integration. of
Oh, there is.
Marek's
Okay.
disease. Yeah, that's right.
But
So-
among human herpes viruses that we know of, this is the only
This
one
is
that
the unique
integrates.
one, yeah. So, so there are a couple of ways that you can acquire this, right? You can get an infection from a virus. You can get non-inherited chromosomal integrations, right? Not somatic cells, or you can get inherited chromosomal integrations, right? And okay, so keep those in mind. Now, the seroprevalence of 6B, as I said, is more than 90%, but we don't know what it is in ancient populations because the only a small percentage is transmitted by germline inherited integrations. And if you want to look at ancient DNA, right, which is the subject of this paper, hard tissue is the most common form of tissue that's available for ancient DNA research. And so you're not going to see the acquired and somatic transmitted versions. You're only going to see the germline integrated, which is basically going to be in all
Because it
tissues.
basically has to end up in teeth or some small bones of the skull, which is what we
Right.
have access to for ancient DNA.
So, what is the tropism of the virus otherwise?
Well, I think it's quite broad, right? You get a rash disease, one thing, and you have all these other supposed lymphoproliferative disorders, so it must include lymphocytes as well. But I don't know offhand beyond that.
I'm looking at Google and it pretty much says the same thing you said too. So,
Yeah.
Okay. So it's people think that the integrations happened a long time ago, and some people wonder whether. So people think 6B is still integrating, but they don't. They think 6A may not be.
Yeah, so the reason they're going to look at ancient DNA here is they want to know where these viruses came from. How long have they been in humans? Certainly, we discovered them in the 80s, but you discover something, it's already in 90% of the population. This was not news. It's been around for a while. And so they want to get a handle on how much has this co-evolved with us and how has it changed? And is it still integrating the same way it used to?
Right. So 6B is still integrating. There have been modern integrations described, but 6A not, and they think it probably just integrated a long time ago. How long, we don't know, but then it keeps being passed on, okay? Which that's what genes do, right? So in this paper, they got the first ancient genomes of HHV6A and B. They have 11 ancient people. Well, of course, they're no longer existing, but their remains. And 2,500 years, they can trace these viruses 2,500 years ago.
They screened something like 4,000 genomes.
Yeah.
Yes, they had
found these 11
4,000
people.
They had 4,000 archaeological samples, and they looked for. They had the sequences had been done as part of another sequencing project, and they looked for HHV6, and they found them in 11 human samples from the 8th to the 6th century BCE. and to the 14th century CE. 6 had HHV6A and 5 HHV6B. So who were these from? We actually know. It's really interesting. Quite a diverse group of people. So there's one sample, OR-D001, is from a female from Ordona in the Iron Age of Italy. EDI-001 is from a male aged 16 to 18 years from the Anglo-Saxon site of Edix Hill in England. JDS-067 is from a male excavated at the Hospital of St. John the Evangelist in Cambridge, England. And 4 originated from a medieval parish graveyard in Belgium. An adult male, an adult woman, and an infant girl were recovered from medieval Estonian sites, 3 different sites. And finally, an older male originated from a site in the Komi Republic of Russia.
That's why this team is from everywhere.
Exactly.
Because their subjects are.
So they're all over the place, and they range from the 8th to the 6th century BCE into the 14th century CE. Okay, so first of all, the tissues where you get these ancient DNAs from are important to tell you about the kind of transmission that occurred, as we were talking about earlier. So for 3 of the samples, the only available, for 3 plus, for a bunch of them, the only available samples were teeth. Okay, so you could possibly find either acquired or inherited infections. But for 2 others, the infections are recovered from bones, and bones are dense and they have not much vascularization. So they say an acquired or localized somatic infection is unlikely. They think these are probably somatic carriage of the viruses, right? And in particular, EDI-001 had multiple sample types, tooth and bone, and the virus could be found in all of them, which they say makes a clear case for inherited germline integration in this individual. So it's interesting that the kind of sample that you use to get the DNA sequence can inform you about whether you had, again, an infection where someone's got a virus from someone else, or someone had an integration in their own non-somatic cells versus somatic cell integration.
So teeth is a really common place to get ancient DNA, right? Is that because it's basically a protected site?
Yeah.
You must be talking about tooth pulp, and it's really, okay.
It's all protected. It comes in a little capsule, and then they bring it in a clean room, right, where there's no other DNA, and they're all suited up so your DNA doesn't get on it, because DNA is everywhere.
DNA, it's really a problem.
And then they drill into the pulp, and they take a little sample out. Yeah, they're very precious, these samples, as you can imagine.
This ancient DNA sequencing,
they have to use Novocaine
really
first, though.
That's right. It's even though they didn't take care of their teeth back then because they didn't know to, they're still preserved because the enamel is pretty tough. Right?
Well, and also because people tended to die by like age 30, so there wasn't really time for tooth decay.
It's funny. The other night on the live stream, some guy asks, Homo sapiens been fine for 300,000 years, which I said, that's wrong, but okay. So why do we need vaccines in the last 80 years? And I said, well, they weren't fine. They were dying at 30. Where would you get that from? And everybody was freaking out over this.
They were dying a lot. In fact, they were dying a lot younger than 30 because that's what, families would have 10 kids and 3 of them would survive.
Right. If you go to an old graveyard, you will see how young people were.
The average lifespan went way up with the smallpox vaccine, mostly because the children survived.
Yeah.
Right.
So as they have pretty good coverage on these genomes. So they have 11 HHV6 genomes, and 9 of them were good enough to be able to compare their sequences with contemporary. There are 278 modern genomes in the NCBI database, and look at single nucleotide polymorphisms and try and put them into phylogenetic groups. And so they could put all the ancient genomes within the known, what they call clades of the modern genome. So they are related to what is around today, and they can give some insights into the integration site, right? You can see if you have surrounding sequence. So basically, they could find that all the integrated 6A clades are represented in Europe by the 14th century CE at the latest, but probably have an older evolutionary history. One clade A2 is represented by a single genome in England dating from the 13th to the 14th century CE. Clade A3 is by one Belgian genome, 7th to 8th century CE, and clade A4, 4 genomes from Estonia and Belgium from between the 8th and the 13th century. And for 6B, their phylogeny yielded termini antiquem, which is a word I had to look up.
Yep.
Antiquem. Did anyone know what antiquem means?
No.
I did not. Not until this paper.
Is a Latin phrase meaning the limit before which. Okay, so.
So this is the latest date that it could be from.
Right. Yes, thank you. And so they yield the
and there's
termini.
also post-quem, termini post-quem, which is the earliest date something can be from.
So first to 6th and 6th to 7th century CE for clade B8 and B5. They could only detect HHV6A genomes in people who died after the 7th to the 8th century CE, which they say is in contrast to the assumption that 6A is older than 6B. And they think that may be sampling biases. They don't have a lot of samples. You need to get more, and I'm sure they will in the future. One of these people was infected by the plague bacteria.
Yes.
Yersinia pestis probably died of that, so they could find those sequences.
The HHV is not what's going to kill you. Yep.
No. So that's the idea here. This is a nice snippet. 2,500 years of these viruses have been around. They have identified at least 8 genomes to be integrated of HHV6, which is very cool, and they can put them in current clades. As they say, most of the genomes are from germline integrated lineages because those are the cells you're most likely to find there, and not circulating viruses or viruses in your somatic cells. And there's one exception, though. There's one sample, KUU029 from Estonia. It had pretty low sequence coverage, so they didn't get much of the genome. But it's the only one that seems to be either acquired or somatically integrated, despite having been recovered from bone. So that's quite interesting. It comes from an infant 10 to 18 months of age, which makes an acquired infection more likely. And that we know today is that infants are the most likely to get this infection.
Well, one of the conclusions I liked is this bit that all the known inherited integrated 6A clades were in the historical populations. So the conclusion from that is it's not integrating anymore. Whereas 6B is jumping in and out all the time.
Yes. Now, this virus could be older. They don't have any older specimens, right? So it may go back even further. But right now, the idea is 2,500 years ago, this is existing. And the somatic integrants, they don't last. But the germline integrants get passed to the offspring. And so what people are getting today from germline is inherited from people 2,500 years ago. Now, the other thing here is that there's no Asian representation in this database. So they say all of our conclusions may be different because this is all European, pretty much.
It's all European. There's no Asian, there's no African, and there's no New World.
Yeah.
So I would be really interested in seeing data from Africa if we can get them, if we can get ancient DNA. Because, an open question here is, did this develop after we came out of Africa, or have we had it since way, way back?
Right.
Yeah, so there was an earlier estimate of the age of HHV6 integration. Here it is. Let me tell you. So the timing of integration is unclear, but one estimate was that they occurred more than 50,000 years ago, which placed it around or before the time of migration out of Africa. Yeah. But we don't know. From this study, we can't go more than 2,500 years ago.
Right.
So basically, this shows that you can use ancient DNA to study HHV6 integration over a long period of history. It was well established before the 14th century CE. It's very cool.
Yeah.
I like that.
Yeah.
I always liked HHV6 because of this telomere integration. So the telomere is a sequence at the end of chromosomes that it's initially made during embryonic development. It's a series of repeats. It's kind of a buffer because then as you age and your DNA reproduces, the ends because of the end problem in DNA replication, right? You can't maintain the ends. They get shorter and shorter and shorter and shorter. But the idea is you have enough from when the telomerase, the enzyme that was active during embryogenesis, made it. And some kinds of cancers, the telomerase is activated and the ends don't get short and the cells keep dividing, right? So shortened telomeres is associated with senescence. It's very cool. And so the virus has this telomere-like sequence in its genome that helps it to integrate into the
telomere, which is also a pretty harmless place to integrate, generally.
Yeah, I feel like it's a harmless place to integrate because there's not much transcription from there. But I wonder, do the cells that have been infected have an advantage because they have somewhat longer telomeres and can have more division ability?
That's a good question. I'm sure it's been looked at. I don't know the answer, though. Hey, I'm getting a phone call, Rich. You should know this, from Zephyr Hills, Florida.
Oh, yeah.
Ever heard of that? You know where that is?
They make spring water.
Oh, you want to sell me some?
That's all I know.
It's a funny
I think
name.
it's probably spam.
I get a spam call multiple times an hour.
I mean, the spam is not made in Zephyr Hills, Florida. It's made in Austin, Minnesota. But that call is probably spam.
Yeah,
spam is made in Minnesota?
Yeah.
Wow.
Austin, Minnesota. I knew someone in graduate school who was from there. And
Austin, Minnesota?
Yeah, I met her and where are you from? Austin, Minnesota. I was like, I feel like I've heard of, and she said spam. Oh.
What's good about spam? Is there anything?
Nothing.
It's cheap.
It's highly processed, right?
Oh gosh, is it processed?
So nobody should be eating it.
Yeah, I think this is, it's basically military rations, right?
Yeah.
But it's a big deal in Hawaii.
Yes.
Really?
Mm-hmm.
Yeah. Spam is really popular. As a matter of fact, you can basically get the equivalent of spam sushi.
Yeah.
So Monty Python made fun of it, right?
Yes.
Yeah.
Yeah.
Rightfully so.
It's popular in all the Pacific Islands that were part of the U.S. occupation in World War II.
Right.
And the GIs showed up and they had all this spam, and it was like some of the first canned food that people had encountered, and it became a cultural thing.
I don't know if I've ever had spam.
You haven't missed anything.
I'm not sure.
You're not sure?
Yeah, Brianne, you'd remember.
Yeah,
Trust
you're
me.
really not missing much except the experience.
All right.
So welcome to TWiV, folks.
Yes.
Yeah.
We just had our first spam arc.
A little diversion between papers, just so you can relax, go get a beverage.
Little intermediate course of spam for you.
Well, in a chef special meal, they set your taste buds between courses with some.
Yeah, cleanse your palate somehow.
Cleanse your palate. Or cleansing your intellectual palate with spam.
With
An
spam.
amuse-bouche of spam.
Or root or stump grinding or any number of other things. Okay, another paper on herpes viruses. This one is in Cell, and this is entitled The Effect of Shingles Vaccination at Different Stages of the Dementia Disease Course. And Alan, please tell us. This is simpler than the other paper.
Oh, yeah, this is a bit simpler. First author is Min Ji. Senior author is Pascal Geldsetzer. They're from Heidelberg University Hospital, Stanford University, Johannes Gutenberg University in Mainz, Germany, Leibniz Institute for Financial Research, SAFE, in Frankfurt am Main, Germany, Cardiff University in UK, and BioHub San Francisco, which I think used to be called the Chan Zuckerberg BioHub.
Yeah.
Mm-hmm.
Now it's just listed as BioHub on this paper.
Yeah, they want to get their name off it.
I guess maybe.
Good. So we have actually covered this a lot last year. In fact, during our year-end review, which was last time, which is, hey, guys, you left me and Kathy
I'm sorry.
all alone.
Yeah.
It's okay. Because, I had said, I want to have 4 people on. That would make a nice discussion. And so we had 4. And then the day before, we're down to 2. But I said, Kathy, we're going to do this. And we had one of my arcs that I picked was herpesviruses and dementia. Now I'm getting another spam call from somewhere else. It's a problem with wearing a watch that has your clothes on.
At least you're not presenting on your iPad when those are coming in. I had one memorable lecture with many spam calls that interrupted.
So we had 4 episodes last year relating to this. So TWiV 1187, anti-herpetic tau preserves neurons via a DNA sensing pathway in Alzheimer's. TWiV 1207, reduction in dementia incidence in recipients of Zostavax, and that was by the same authors as this paper. TWiV 1249, herpes simplex virus infection doubles the risk of dementia. And TWiV 1263, varicella-zoster reactivation and the risk of dementia. And so why herpes viruses? Well, they're neurotropic, and we know that neuroinflammation plays a role in dementia. So we're going to look at that. And as you have seen, several findings support the idea that herpes viruses are involved in dementia, as I've just told you, and we covered last year. So, herpes zoster is one of them. And vaccination, as we saw in a previous study, can reduce the incidence of dementia. And just to remind you, in these studies, they took advantage of a randomization that happened in Wales and then later in Australia. So the UK National Health Service, when they rolled out the live attenuated herpes zoster vaccine, that's called Zostavax, made by Merck, they used strict date of birth eligibility rules. And so you can compare the incidence. So of September 2nd, 1933, if you're born before there, you can get zoster. If you're born after, or maybe it's
if you're born after that, you can get Zostavax. If you're born before that, you don't.
So it's a perfect cutoff, and you have 2 populations that should be similar and not have a lot of confounding differences.
And probably the logic for that is if you're older. So these are octogenarians we're talking about at the point when this was implemented. If you are older than that, you probably don't need to worry too much about shingles.
Right.
You've already had it, or you're not going to get it, or whatever, and something else is going to get you before you have another outbreak of it. But you do want to start somewhere, and so they set this cutoff date and said, okay, everybody under this age is eligible for this vaccine.
And that was before we knew of the connection between dementia and the vaccine. But now anybody should get vaccinated, right? Because dementia is going to be a major contribution to deaths of older people. Right.
And so all of the papers that Vincent mentioned earlier are some of those examples of sort of more recent studies that have been able to really give us insights on this link that we couldn't do before without really large numbers of subjects and a lot of data. And so that's sort of that difference between HHV. I wanted to put way too many H's in there. 6, where we don't know nearly as much. And this is sort of, okay, here's another one that's carried by 90+% of people. But now we have enough data to actually know about this pathology.
Yeah, so and another reason you need a huge data set for this is because dementia is a real problem to look at because it takes so long to develop. Mm-hmm. And so you have to find some kind of a data set that extends far enough that you can track people and see who developed it and who didn't, and who got vaccinated and who didn't. And that's what we have here.
So it's basically a longitudinal study. It's not retrospective, which is good because you're following people with time. So in the study in Wales, they found, which we talked about last year, that having a vaccination, herpes zoster, prevented 1 in 5 new dementia diagnoses over a 7-year follow-up period. So it's substantial. And then they subsequently found the same in the Australian study. So what else do we need to know? Well, we'd like to know when this vaccine has a benefit, because in that other study, they just said dementia or not. So they had two aims in this study: to determine the effect of vaccination on new diagnoses of mild cognitive impairment. So you start with people who have no cognitive impairment, you vaccinate them and see what happens over time. And then look at deaths among people who already have dementia and see if there's any effect of the vaccine. Right?
Yeah, so they
two
previously
options.
looked at dementia diagnoses. Now they're looking at the extreme
The
ends,
two ends, yeah.
mild cognitive impairment and death, and seeing if we have an effect on those as well.
So the same data set in Wales, and again, you have the September 2nd, 1933 date of birth eligibility threshold. And so they first do statistics to make sure, because they have data on the threshold, people who were eligible or not, and whether they received it or not. They have to make sure, right, if someone was eligible, that they received it, because not everyone who's eligible is going to receive it. And then they have individuals without any record of cognitive impairment before the start of the vaccination program, and then they're eligible, and they're born after September, and eligible vaccine receipt. So they had an increase in the probability of ever receiving the vaccine. So you may say, why did you look at that? As again, you have to make sure they got the vaccine. So they're looking at getting vaccine as a statistical thing. Okay, so the increase among patients living with dementia on the start of the vaccination program was from 0% to 28.7%. Okay, so these are the people that they followed them for 9 years, I believe. All right, and we'll come back to possible confounding effects a bit later, but because of this strict cutoff, these populations are likely to be very similar, right? Okay, so among individuals with no record of impairment prior to the vaccination program, 20,000 were newly diagnosed with mild cognitive impairment during the 9-year follow-up period. All right. And compared to those who were born after the date of eligibility, so they're eligible for vaccination, it decreased the occurrence of new diagnoses by 1.5 percentage points. And if you scale that to the number of individuals who got vaccinated, if they were eligible, the effect of actually being vaccinated was a 3.1% percentage point reduction in new diagnoses over 9 years. Okay.
Yeah. So basically, when they looked at the two different groups, they saw that the vaccinated group had a 1.5 percentage point decrease in getting mild cognitive impairment, but only about half of them had actually been vaccinated.
Right.
So that you can say that the vaccination could lead to a 3%.
Right. Okay. And then they looked at deaths due to dementia. So in this study cohort with people who had dementia prior to the start date of the vaccination program, 7,000 died. That's 49% of the population died due to dementia over this 9-year period. If you were eligible for vaccination on September 2nd, that decreased the occurrence of death due to dementia over 9 years by 8.5 percentage points. And if you actually received the vaccine, you had a 29% reduction in deaths over 9 years. That's a lot.
That's a big number.
Yeah.
That's big.
Because you can die from a lot of things.
Yeah.
And 30% is
That's
a lot.
a big number.
Get zoster vaccine. And by the way, this is done with the live attenuated, but everyone gets Shingrix now, the inactivated. And
Right.
We don't have...
They
I think
make
a
the
paper
point somewhere in the beginning of this that
Is it
you sort
inactivated
of
or subunit?
Subunit, sorry.
It's subunit.
It's a glycoprotein, yeah.
Sorry,
What do they,
Rich.
sorry, Rich,
that's
what do
okay. They
of-
make the point somewhere towards the top of this paper that the live attenuated vaccines seem to have, in terms of protective effects like this, seem to be better than subunit vaccines. I think that remains to be,
It remains to be
verified,
seen in
but-
this particular case, but I think they were talking generally about additional beneficial effects of vaccines in
Right.
general, which occur more commonly with attenuated vaccines than with
Yeah.
others. But in this case, we don't actually know because the rollout of Shingrix was relatively recent, and we don't have the data to do that kind of an analysis just on Shingrix.
Yeah.
This is like a one-time experiment.
This
right?
is a one-time experiment, yeah.
I think there was a paper last year, we didn't cover it, that looked at Shingrix and found also reduction in dementias, but we didn't do that one. So then they want to know, okay, so this date cutoff is great to eliminate confounders, right? Because what's the likelihood that on September 2nd, 1933, you're going to have something happen to some members of the population that's going to make this difference, right?
Right.
So that'd be another kind of intervention. So they looked at this with their data. So first, the September 2nd threshold only had a significant effect on that vaccination program, but not in any of the 3 years before and after.
Yeah.
So in other words, that going 3 years in either direction reduces the probability that an annual intervention existed, right? So people are having something else treated, and that influenced their...
September 2nd is the day to get your whatever, right?
Yes.
Yeah. So I really thought I thought a lot of these controls were pretty clever. And so this idea of, well, what if we take the data and do a cutoff of September 2nd, 36, 35, 34, 33, et cetera, and show that 33 is the only year that matters, was pretty good.
So then do we observe systematic differences across the September 2nd date birth threshold in preventive health services? You know, did certain people get something and not others? They didn't find any. There were no differences in the incidence of new mild cognitive impairment diagnoses or deaths on either side of September 2nd, right? And finally, other than dementia, they didn't find any other effects of this September 2nd birth date eligibility. They looked at 10 of the most common causes of daily annual life year expectancies.
Disability adjusted life years.
Yes.
Yeah.
So this does not affect your risk of heart attack or cancer or
anything
anything else.
Right. Now, one thing that's really interesting is most of this effect is on women,
Yes.
not men.
In fact, the entire statistically
Yes.
significant effect is in
on
women.
women.
When they break it
And
down by sex, they find men, there may be a difference, but it's not statistically significant.
Yeah, so there's a little difference, but it's not significant, and it's mostly women. Yeah.
I found that somehow surprising. Okay. Though, you can make up, and they do, lots of reasons for it.
Yeah.
And it makes me think of, the recent, I don't know what the current situation is, but the last few years in study section, and grant writing that I experienced, they made a big deal about making sure that in studies in humans and animal models that they were gender balanced. Okay. Because there's been so much research historically that's been biased towards males in particular. And this is a wonderful justification, in my mind, for that attitude, to have these studies gender balanced, because this is a huge effect.
Yeah, I'm always impressed by sort of the size of the effects in a lot of these studies, actually.
Yeah.
Now, in their previous, if I recall correctly, in their previous study looking just at dementia diagnoses, they found the effect in both genders, but it was more pronounced in women. I think it was in both, but skewed female. And this one, it's just overwhelmingly female.
They also wanted to see if the vaccination could help people with more advanced dementia. So they divided their population into those living with dementia at the time of the start date of vaccination, into those with more versus less severe dementias. And they used some indicators of dementia severity to do this. For example, the risk of all-cause mortality, the total number of days spent as an inpatient, the number of hospital admissions, the number of times a diagnosis of dementia was recorded, risk of death, etc. And so they found that tentative evidence for a protective effect on vaccination and deaths due to dementia, but it's greater for those with more advanced dementia than on people with less severe dementia. Okay, did I make that sense? So
Yep.
if you have more
Yeah.
severe dementias, you're more likely to be helped by being vaccinated.
Yeah.
And then they did to look at what kinds of dementia are most strongly affected by vaccination. They mostly looked at women because the effects are largely driven by this group. And they looked at 3 common types of dementia: Alzheimer's, vascular dementia, and mixed dementia. And so the reductions in dementia incidence from vaccination were largest for mixed dementia, and the others were lower. It's interesting. Also, people, they want to know about mortality, all-cause mortality in patients living with dementia at baseline, right? And so vaccination decreased all-cause mortality over the 9-year follow-up by 6.5%, which is not bad, right? Actually, that 6.5% is just being eligible for vaccination. In the people who actually received vaccination, is 22.7% reduction in all-cause mortality over 9 years.
And is that gender skewed as well?
They only looked at women in these.
Ah, right.
Yeah.
Right.
Well, that's right. They didn't have enough data for the men or something like that.
Well, the men, they didn't find statistically significant
Right.
effects, so now they're looking at the group where
Right.
they see
Okay.
Right.
the big effects.
But
Well, actually, they did for deaths. They did look at both, but it's zero. They said it's basically statistically zero among men. So these 22.7% reduction in mortality is mainly in women. Yeah.
and we've sort of been talking about this a little bit, but Vincent just mentioned when he reported the previous piece of data, what the 6% was for being eligible for vaccination. And then the other number was for actually getting vaccination. And it's really important that they did this study in terms of the being eligible for vaccination and didn't just look at those who were vaccinated versus those who weren't. Because if you just look at those who are vaccinated versus those who aren't, you can have more confounding variables. You could say the people who are vaccinated have different healthcare utilization, or the people who are vaccinated, have other types of differences that might correlate with their dementia, maybe education levels, which might also be involved with dementia or something like that. And so the really unique part about this is that because of this strict cutoff date, they can do the part of the study where they just looked at eligibility, where you don't expect to have those same kind of confounding variables.
Right.
They also looked at non-dementia associated deaths, and those are not affected by vaccination. So that's a nice control.
Yeah.
So those are the data. Live attenuated herpes zoster vaccination reduces both new diagnoses of mild cognitive impairment in people that don't have any record before, and it also reduces advancing impairment and deaths due to dementia in people that already have it.
So they extrapolate from that to say that it doesn't matter where in the course of dementia you might be, the vaccination is going to be of value.
Yeah.
And they make it has not escaped our attention statement. They say the finding that herpes zoster vaccination has a beneficial effect on the dementia disease process would be of great significance to population health, clinical medicine, and dementia research. Yeah.
No kidding.
Yeah.
Yeah.
Certainly would. The protective effect larger among women than men. They also found this in their previous whales study, as Alan mentioned. They don't know why, but they say maybe there's immunological sex differences playing into this. I think women are more frequently getting shingles. That may be part of it as well.
Yeah, I mean, the very, very, very broad data that are out there tends to support the idea of women having more immune activation, and thus higher propensity of autoimmune disease, higher propensity of sort of inflammatory pathology, less pathogen-induced damage. And so if what's happening here is we're getting excess reactivation, then the women are going to be seeing more of that, more immune damage coming from containing that activation.
Right.
So they say, of course, we need to know the mechanism here. We don't really know. And there's some evidence that varicella-zoster reactivation is associated with more dementia. We talked about that paper last year. And they also say that reactivation of VZV may lead to reactivation of herpes simplex virus in the brain. So you're getting a double hit. So preventing herpes zoster effects is good for preventing the herpes as well. So there you go. I think that's really good. And it's continuing. I mean, this is cool. We picked this arc for last year, and it's continuing. I like it very much.
Yeah. I just think that this whole arc is making me think a lot about sort of the long-term future with VZV. And this might be an age bracket thing. But, you know, I had chickenpox when I was 4. But I know that many of my students have never had chickenpox because they were vaccinated. Of course, they were vaccinated with a live attenuated. Yeah. And so there might be this question of, do they have latent virus? And could they eventually progress to this? But is there a difference in whether or not I'm subject to dementia versus those people who were not actually infected, who only got the live attenuated vaccine? And further, if in some future time one were to switch to using a subunit vaccine, and people were not getting that latent infection, how would that influence sort of dementia on a population level?
If we eradicate chickenpox.
Yeah. I find this whole idea really kind of fascinating. I think it's tricky because I'm not sure that increased vaccine recommendations are going to happen right away.
Yeah, right.
But I think that the idea of how could we actually sort of impact dementia with this vaccine that people hadn't really thought about as a vaccine thing, and you know how much we have actually been influencing dementia since the 90s in giving kids a chickenpox vaccine that we didn't even know about. It, to me, is just fascinating.
Yeah,
I think eradication is a problem because you'd have to prevent infection, and otherwise it gets latent, right?
Yeah.
And so it's the same problem with HIV.
Yep.
I didn't actually mean we would eradicate it, but just as chickenpox rates plummet, which
of course
they're doing, then what happens 70 years from now?
Right. Yeah. Well, and children still get Zostavax, right? Children are still getting the live attenuated, and it's just the elderly population that gets the subunit.
Yeah.
Elderly.
So we'd also have
Thanks.
Yes, that's right.
Sorry,
kids these days.
Over 50.
It's just the older than me population.
I was
feeling really left out.
Over 50.
Yeah.
No, so they say the best ways to prevent dementia are exercise and use your brain. And now we can add getting a herpes zoster vaccine. Vaccines make America healthy again.
Yeah.
Can you imagine? Hey, RFK Jr., get your head out of the sand.
I know. See, this makes me again say, why can't 40-year-olds have this vaccine?
Actually, it's being trialed in kids, the subunit zoster vaccine,
but,
Shingrix.
But why 50? Why not 40? Why not 30? Like
because most shingles occurs over 50.
Yeah, but you know,
Well, they didn't. They
But I want to
didn't
prevent
know.
dementia.
Yes.
Well, it used to be that, right? Zostavax was the same for kids and for zoster, wasn't it? Or,
Yeah, there was
Yeah.
the shingles vaccine was basically a double dose
Double
of
dose, okay.
Zostavax.
Yeah. But the 50 is just, most of the cases occur over 50. Although I had one student in the class one year, and she was 20 something, and she had gotten shingles already, so it can happen. She was immunosuppressed, so it can happen. Okay, let's do some email.
Goody.
I'm going to take this first one because it has to do with microbe.tv. So last time I read an email from someone who said MicrobeTV is not working. I can't find the most recent episodes. So I read it just to tell everyone we're having some issues. And Portia writes, just finished TWiV 1285. You mentioned the website not updating properly for some people. I was one of those, but when you said it works for you, I got a thought. I tried it out, and this is what I found. The problem is not the website, but the browser. I was on my phone using Safari and not seeing the latest episode. So I said, why not try Chrome, which I never used, and it worked fine. So I cleared the browser in Safari, and it worked too. Then I tried the same on my computer, and it worked after I cleared the website history for MicrobeTV. So if you're on Safari on a Mac, go to Settings, Privacy, select Manage Website Data, and remove the data from MicrobeTV. On my iPhone, I went to Settings, Safari, whatever browser you use in the apps, and then select Clear History and Website Data. I don't think you get to choose which website, so all your cookies and history will be cleared. It's all right. You shouldn't eat cookies anyway. After that, the website showed the updated episode. Thank you for all in your co-hosts do. You feel like family after all these years.
I actually have
folks,
my browsers configured to flush the history and cookies every time I close them.
Really?
Ooh.
Yeah.
What browser are you using? Firefox?
Well, I use Firefox on Linux on my desktop, and then I do use Safari on my iPhone, but I created a shortcut button to flush the history because I had found this problem on other websites where it was like, oh, I got to flush the history in order for this darn thing to work. I said, okay, I'm just going to have a button for that.
So you don't have a problem with MicrobeTV then?
No.
You see all the latest episodes?
No, it loads fine.
I was having a problem in the fall.
I mean, flushing your history and cookies also clears out all the trackers and everything that you've picked up by walking through the brambles of the internet.
Brambles of the internet. I like that. Indeed, you get cut by them, those brambles, do you? Okay. So we have a short and a long one. So Alan, take both of those.
I'll take both of these. Simon writes, now RFK Jr. has personally removed mercury from all the vaccines and updated the vaccine schedule to one he approves. Will all the anti-vaxxers follow him and ensure all their kids are vaccinated as stated in the new CDC childhood vaccine schedule? But I know the answer. Ask Paul Offit and Daniel.
It's a really good question, right?
Yeah. No,
no, they're going to say he's copped out.
Right.
Right.
No, these are extremists you're dealing with. Yeah. Next up, Jane writes, Dear This Week in Virology staff, your podcast was recently brought to my attention by a former student of mine, Jessica Alvarez. Jessica lived in Hopkinton, Rhode Island most of her life, but is now a lab scientist in Madrid. She was an extraordinary student and has always been an inspiration to all who have known her. I am grateful to have had her in class so many years ago and happy that she has not only excelled in her professional life, but that she made me aware of your podcast as well. She happened to mention that you often discuss the topic of getting young children interested in the field of science. I spent the majority of my educational career focusing on just that topic. Much of my career in education focused on developing STEAM, science, technology, engineering, and math, materials for science. What is the A for?
Art.
Arts.
Art. Okay. So basically, that's the whole curriculum, isn't it? Anyway, when creating a science curriculum for the elementary grades, I always loved the challenge of not only making learning fun, but engaging as well. One area which seemed to be lacking was that of supportive reading materials for understanding basic science concepts. Many books were either too technical, had vocabulary which most children could not comprehend, especially English as a second language students, or were just too dry in content. Recently, I published a children's book titled Have You Seen the Berry Monster? It's written in the format of a science-based poem for research, For research has shown children retain more when reading via a rhyming pattern. My hope is that this colorful and engaging inquiry format will provide parents, grandparents, and teachers an opportunity to further explore animals and their habitats, the environment around them, and the concept of food chains in a way that makes exploring this topic fun. Perhaps it'll also encourage children to explore the outdoors more often and get them excited about life science. Have You Seen the Berry Monster will show children that they are already scientists, for they make observations every day. In addition, through their use of inference and inductive thinking, they will come to solve this mystery. Now, after 34 plus years in education, my wish is to continue to support science education by donating proceeds from the sales of this book to science literacy in the elementary schools. Therefore, I hope you will provide me with the necessary information so that I may send you an ebook version to review. Book is available in an 11 by 8.5 hard copy and as an ebook via Ingram Spark. Gives the ISBN number. Perhaps it will provide you with some food for thought. Best regards, Jane Carlson Pickering, who is the author of Have You Seen the Berry Monster?
She's got a website with this book on it.
Nice.
Yeah.
You know,
very nice.
It's
Send it
very
along.
funny. Last week, my pick was a book that I had as a kid called Science in Your Backyard, okay, in your own backyard. And I have the hard copy here. It's 1958, and I showed it. And Kathy also had the same book. It's about
cool.
going in your backyard and just making observations. So she goes through different chapters and say, you can make notes of what you see and what's going on. And that's exactly what she's talking about here. Get them excited to explore the outdoors and all that stuff. So it's really cool.
Oh, good.
Coincidence. Brienne, you're next.
All right. Alyssa writes, thank you so much for all you do to educate the public. I've been listening to your podcast since the start of COVID and have been a regular listener since. I live alone, so I have no worries about anyone bringing home any viruses. I always wear a mask when I go inside anywhere and I get vaccinated. So far, I have not gotten COVID or any other respiratory illnesses. I have a question. I have noticed these wearable air purifiers. They claim to have FDA approval, but with the state of the FDA, I don't believe anything they say. Are they as effective at preventing the wearer from catching a virus as well as mask wearing? If so, which one would be the best? I've listed 2 that I noticed on Amazon below. And she gives us, she lists 2 of these air purifiers and says, again, thank you for fighting the good fight, Alyssa.
I don't know if you've seen these, but these just hang around your neck, right?
Oh.
And they dangle on your chest. So
Yeah. I'm not buying it.
I don't
So
think
first of all, I looked it up. They're not FDA approved. Okay. And I don't.
Also, I want to clarify something about FDA approved. If it's not a device that's regulated by the FDA, then of course it's approved. Like if you didn't try to get this approved as a medical device, you're basically saying the FDA doesn't care that we're selling this.
I see.
Right.
Right.
I mean, yeah,
so it's not FDA disapproved.
It's not FDA disapproved, right? You could probably make some claim in that vicinity and not get called out.
I just don't think something hanging around your neck
No.
is going to do anything.
No.
Wear
I'm
a
not
mask.
buying it.
Yeah.
Just by glancing at it, I'm not believing this. Unless they're going to show me the data, but I don't think so.
Rich, you're next.
Anthony writes, let me see. He's got a link to a YouTube site called...
It's TWiV 1285, and he's quoting from that episode.
Okay. 1285, TWiV 1285 at 1:45:07. Have you heard or seen this Chinese virologist named Li Meg Yan? Early in the pandemic, she secretly left China and walked out on her Chinese virologist husband and was paid off by Trump. Oh, by the Trump administration. I remember this. By the Trump administration to come to America and espouse her lab leak theory. There are ties to her being paid by Steve Bannon $10,000 a month to keep her here and spread a lab leak theory. Now she's gone quiet and missing. Concerning. Oh, and then concerning another link to this same thing. Ling Mi Yan worked at a prestigious lab at the University of Hong Kong. It's obvious why Li Mingyang did it. She's crazy. Her mental illness and the people taking advantage of her has ruined her life, her parents' lives, and her husband's. The real question is, why do thousands, millions of Americans listen to her? It takes abject ignorance concerning China to think that someone in Hong Kong would know anything about Wuhan. People in New York City would laugh at the suggestion that a bank teller in Midtown would have any idea of the goings-on in the stock market and Wall Street 5 miles away. For her mental illness not to be recognized immediately for what it was, for what it is, but instead to resonate, requires a corresponding lack of balance in the audience. For the anti-vax Maha crowd, similar examples of hysteria or delusion can be compiled into a long list. The strange agitation about children wearing masks, statements about Bill Gates setting up a secret bioweapons facility in China. A fourth grader seeing this in a comic book should recognize it as nonsense. Dr. Hotez's project to educate the general public about vaccines and autism has resulted in death threats, people saying that more have died from the vaccine than from COVID-19. There's not enough room in the margin to complete the proof. The tsunami of warped thoughts needs a psychoanalyst probing. The rock-solid ignorance is nothing new. An African immigrant told me maybe 7 years ago that he was frequently asked, "Who is the president of Africa?" Sad to relate, the general cultural illiteracy was bad and has been getting worse over 50 years. That's a general problem. That's a serious problem. As just one core example, none of the new residents, young and educated, that I've encountered in the New Jersey Heights overlooking Hoboken and the Hudson have seen On the Waterfront. No idea who Marlon Brando was. Didn't bother to ask about A Streetcar Named Desire. Going on a half century ago now, there was a local college that made news each fall for a questionnaire giving to incoming freshmen. Most were completely ignorant of history and geography. Examples: who was Himmler? Most popular answer: the inventor of the Himmler maneuver. This was from a time when every eatery in New York City had a poster on the Heimlich maneuver. Napoleon, Washington, Caesar, placed in chronological order. Results no better than chance. What is Vietnam? Most had no idea. For what it's worth, Anthony.
Oh my.
Jeez. Nice rant, Anthony.
Thank you.
Yes.
That's a good one. By the way, everybody needs to see On the Waterfront. I could have been a contender.
I could have been a contender, yes.
All right. John writes, measles coming on strong in South Carolina, where there happens to be an MD, Annie Andrews, up against Lindsey Graham. She herself says she never imagined that her campaign slogan would be, quote, "It's me or the measles," end quote. In haste from a sub-zero C Greater Braddock, which is expected to hit 15 C this coming Friday. Who's next, Brienne? I don't remember.
I think it was Alan next. I mean,
Am
I'm happy
I?
to go.
Yeah, I went after Vincent.
Okay, go ahead.
Volker writes, Dear Vincent, I wanted to follow up with two brief points you raised relatively recently during the clinical updates. I do have a bit of a backlog at the moment, and I'm not sure this is an ideal email to read on the show, but I'd appreciate a short reply if I've misunderstood anything. TWiV 1270, at 40 minutes 14 seconds when discussing the Canadian Paxlovid study, you say, well, the thing is, Daniel, this is an observational study, right? And you can only make associations, you cannot make conclusions. The conclusions are drawn from the double-blind randomized clinical trial, which was originally done by Pfizer and showed this bigger decrease in hospitalizations. So those are the data we should look at, not an observational study. Makes no sense. I was a bit surprised by this framing. Is the implication that evidence from observational studies should be largely discounted here, even though the RCT data, randomized controlled trial data, come from a period when essentially no one was immunized, whereas today immunization is nearly universal? I'm asking especially because observational studies are commonly relied upon when evaluating or refuting alleged associations between vaccines and autism.
Why don't we do that one first?
Yeah, go ahead and answer that one.
So I would say we don't discount them. But as we just discussed in the observational study on zoster vaccines and dementia, it's hard to make conclusions because of confounding variables. Now, the study we just did is beautiful because of the design, right? That cutoff date can help you eliminate confounding variables. The other way is to have very large studies in multiple places. And that's the case with the vaccines and autism, right? You have dozens of studies, or over a dozen studies, all coming to the same associative conclusion. And then because you can never prove that something doesn't do something, right, in an observational study or even in a randomized clinical trial. So that's what I meant to say, that people were in many observational studies, they don't account for confounding variables, and then it's useless.
So when I was teaching epidemiology this past semester,
right,
one of the figures in the textbook, which I use very, very, very frequently, was a little pyramid that listed different study designs and sort of how strong their evidence, how strong of an evidence for causation they said. And none of them actually gave you full evidence for causation. None of them could completely prove causation, but some were sort of stronger than others.
Mm-hmm.
And an RCT is sort of higher on that pyramid than some of the other types of observational studies. But that's assuming that there's no other difference. Like, as you say here, the percent of the population being immunized. And one thing that even comes in stronger than RCTs is data from multiple different studies sort of combined going towards the same thing, like Vincent said, which is really often what we're seeing with those vaccines and autism situations, is that we're looking at the results of many hundreds of studies, not just the one study. And that ranks even higher than the RCT.
Okay.
Okay.
Go ahead, yeah.
Continuing the letter, TWiV 1272, at 10 minutes 15 seconds, referring to poliovirus detected in German sewage, you say, but it could be that it's even circulating in Germany because they use inactivated polio vaccine and the intestine is still susceptible to infection. So it could be that it is circulating. As long as you keep immunization rates high, it doesn't really matter. This seemed somewhat at odds with what I understood from episode 1243, where, if I recall correctly, you, or rather the person who shall not be named, indicated in response to a question of mine that there was no substantial difference between IPV and OPV with respect to gut immunity. Wait, person who shall not be named, was that Amy?
Yeah, now we can say
we can
Amy,
name her. Yes, okay.
because she's no longer at
Because
the
she's
FDA.
no longer working for the government. Apologies if I'm missing something there, but TWiV is an important pillar of my worldview, so I try to resolve apparent inconsistencies when they arise. Best regards and Happy New Year, Volker.
Well, it's true, as Amy said, there's really not much difference in intestinal immunity between IPV and OPV. So, but what I meant is that they are immunizing at all, and it doesn't matter which vaccine they're using. They're using
Right.
IPV. You're susceptible, and that's how these viruses are circulating. So I didn't mean to give the impression that IPV is worse than OPV in intestinal immunity, because as Amy has said, there's no difference. Yeah. I hope your worldview is okay now, Volker.
Yes.
Yeah.
I don't want to
Make
mess
sure
it up.
Volker's straightened out.
All right, last one is from Brianne.
All right, Charles writes, hello, Twivers. The weather in Chapel Hill is cold and windy, 39F, 4C. At least it's dry today. Pre-pandemic, if someone said turtles all the way down, I thought they were talking about a Hindu creation myth. Today, I am not so sure about that. There is an anti-vax book called Turtles All the Way Down, Vaccine Science and Myth. It is full of pure male bovine excrement, which seems to be what anti-vaxxers want. I see it quoted frequently by the misinformed. Later,
Wow.
Charles.
It's so bad. That turtle's so sad. That turtles all the way down gets associated
Yeah.
with
Yes.
the anti-vax movement.
I mean, I associate with the Dr. Seuss book, right?
Yeah.
Which is the Earth is sitting on turtles, and what's under
Well,
the first? It's turtles all the way down. Is that
Yertle the turtle. But
Yertle the
this
turtle?
The phrase goes back to an alleged quote from, I think, JBS Haldane, who somebody, it was some science popularizer from the 19th century, and a woman confronted him on the street about the Earth being round versus flat, and she claimed that it was flat and supported on the back of turtles, and he, of course, replied, "Well, and what are the turtles on?" And she replied, "Oh, you won't get me with that. It's turtles all the way down."
That's funny.
Yeah.
There's a whole Wikipedia article on this.
I'm on the same article.
I'm looking at it. And there's a background in Hindu mythology as well,
Yeah.
apparently.
And not to mention, I have to mention Discworld, of course, which is turtle all the way down, for those who get the reference.
I use it now and then.
Yeah.
I don't, for other purposes.
Any kind of recursive infinity that,
it's turtles all the way down. How dare they use that?
Yeah.
bastardize that wonderful
that's
phrase. Yeah.
All right, time for some picks of the week. Brianne, what do you have for us?
So I have something that just came across a education email site that I'm on, or email list that I'm on. I've known about this project for a while, but they've actually put together a full book that is linked here as an ebook, where there are a group of people who are doing annotations for TWiV, or This Week in Microbiology episodes, to align with something called the Vision and Change recommendations, which are some AAS, AAAS recommendations for science education in biology. So there are certain statements that people sort of think of as key learning objectives in biology. And this aligns the papers and sort of that are discussed on different TWiV episodes with those, and has some resources and activities that could potentially be used to use those TWiV episodes in microbiology classes. So I know of some scientists who've been working on these annotations for a while. I've used some of their previous works of this, but this is a full book with a huge number of episodes. I've used some of these with my microbiology class because it's a great resource and means that I don't have to go through and re-listen to every single episode to remember what's discussed where. I also have used a lot of these as models for other, say, TWiV or immune episodes that I wanted to use in class. So this is a fantastic resource if anybody is looking for microbiology related resources using TWiV for a micro class. I can't say enough to thank the group of people who have done this.
This is amazing.
This is awesome.
It's very cool.
Wow.
How many TWiVs are there? This page of transcripts goes up to 104. There's here we go. 333.
348 we recorded yesterday.
Yeah, they've been doing this for a while, and it's not all of the episodes. I think it's something like a team of like 50 different people, and they've done annotation at this point for 99 episodes.
Wow.
It's very cool. It's a nice execution of, our original goals to teach people science through these podcasts. It kind of formalizes it, right? They annotate the podcast, they make transcripts and so forth. I like that.
Yeah, it for me, I found it a really nice way when I've wanted to assign, because I assign episodes for a bunch of my classes. I have some assignments that I use a lot. And I like this when I'm looking at micro episodes to help me pick episodes that will get to different topics I'm talking about at different times. It's an easy way for me to say, oh, I need an episode about this. Oh, here's a link and here's the annotations all ready for me.
Yeah, and this organization is great. I mean, you can look at structure and function, and you get a list of a dozen episodes that you can, metabolic pathways or something. And
those are the vision and change categories.
This is great.
They say the podcast is accessible to both novice and advanced students in biology. Students in introductory and advanced micro courses can benefit from the scaffolded podcast listening experience, following along with the host reasoning and discussion, as well as the host introductions to new concepts and technologies. What does it mean, scaffolded?
So scaffolding is a term that people talk about with education, where you kind of break down the steps of something. And
I
so
see.
instead of just saying, here, do this huge project first, you might say, okay, let's work on this skill and this skill and this skill. And then you're going to put them all together for the project.
Cool. Well, we're scaffolded.
Yes.
This would be good for all the podcasts, actually.
Yeah,
'cause,
I mean,
Yeah.
it's hard to find things, right? Like
Yeah.
searching is hard. And even in a year, like we did our year-end review, I had to go through each one at a time because searching isn't good. But if someone annotated them, that would be awesome. And just index them, if anything, by topic, right? That would be cool.
Yeah. So this sort of thing, I know how useful I've found it, so I thought that others might find it useful. And yes, if it was available for TWiV and Immune, I would use it even more. But
Yeah.
that's okay.
Feed all the podcast to an AI machine with
No.
instructions. No,
No,
It would screw
I
it up.
wouldn't trust AI to get
it would
this right.
screw it up. Yeah.
Yeah.
You'd have to go check
What
it
you
out.
would like is just a list of the papers discussed on each podcast, right?
That would be
Yeah.
handy.
That would be handy.
That
all
you
by
could
itself.
do with code, probably.
Because the problem is we have all the episodes listed, like 5 per page, and you have to keep
Yeah.
going through the pages, and it takes you a long time.
Yeah, but I mean, it makes it easier because all the titles make sense, right?
Yeah, that's right.
Right.
That's right. It's perfect.
Our
All you
titles
need
are
to
always
do-
so intuitive.
Exactly.
But at
Just
least the
look at
papers are
title.
linked. So when I will put together assignments, it's easy enough to find those specific papers
Interesting.
from the
So-
links.
Yeah, so that's
You wouldn't
I found
need
that.
AI to pull out the paper references. You could, if I had time, I could probably write a Python script for this. Just scrape the pages for the links to the papers and compile them all and then link them to the episode that they're on.
So it could
Anyway.
distinguish between links to papers and other things.
You could probably write some regular expressions to do that.
Well, that's very cool. I'm glad that these people are doing
Yeah,
this. It's
very
nice.
cool.
Thank you very much. All right, Alan, what do you have for us?
Okay, so I'm going to be picking a lot of books coming up. They're not necessarily all going to be book picks, but I realized in 2025 I read something like almost 60 books. Been doing a lot of reading, so expect a lot more books coming through. Anyway, this was one of my favorite reads from last year, and I gave it as a Christmas gift to 3 people. Starter Villain by John Scalzi. This is 2023 book. Scalzi is best known for sci-fi, especially space opera type stories, always with some kind of unusual twist from what you're expecting for typical space opera. But this one is actually set in contemporary times, a little bit of a different thing for him, and it is wonderful. It is hilarious. It's a wonderful satire. You start off the main character is this guy Charlie, who's a business reporter who got laid off from the Chicago Tribune because of what's going on with newspapers. And now he's working part-time as a substitute teacher. He's living in his parents' house back in his hometown. He's basically failed at life in middle age. And then a rich uncle he barely knew dies, and it turns out Charlie has inherited his uncle's supervillain business. He didn't realize his uncle was a supervillain, but now Charlie's in charge of the business, complete with a volcanic island lair, patrolled by intelligent talking dolphins. And it is wonderful. Just send-ups of the stupidity of our present moment, and very, very well done. And Scalzi is a fantastic writer. It's a page turner. I mean, you could finish this in a couple of sittings.
Been
Highly
in my
recommended.
to read pile for a while, and you just moved it up much closer to the
Yes.
top.
And the cats are in charge.
Yeah, I like the cover. It's
The
great.
cover is good.
Very
Yes.
cool.
So speaking of cats now, we did a paper on TWiV yesterday showing that when kids have dogs during their adolescence, they grow up with fewer problems, fewer social and neuropsychiatric problems. And they found that with cats, it goes the opposite way. They have more problems if they have cats. The reason it was on TWiV is because the dogs actually transfer their microbiomes to humans by licking and kissing. And they did an experiment where they took saliva from kids who had dogs, and they put them into mice, and the mice had better social interactions and all this stuff.
Wow.
Very interesting. But I thought it was so funny that these cats had the
Yes.
opposite effect. Okay. Rich, what do you have for us?
So I got another book by John Scalzi. We're on a Scalzi arc here. I actually had another pick that I was going to post that I'll do another time, but then I went on to the doc we use, and I saw that Alan had a Scalzi pick, so I thought, let's make it a Scalzi pick fest. This is a novel called Red Shirts, which is in the space opera category. It's called Red Shirts, a novel with 3 codas, and 3 codas is significant. This was I think I picked Old Man's War, a while ago.
Yeah, you did.
And I've read all 3 of those now, the whole trilogy. So I'm on my own little Scalzi run here, and I'll have to read Alan's pick. But at any rate, my son knew that I was on a Scalzi kick, so he got me Red Shirts for Christmas, which I devoured. Those of you Star Trek folks out there will recognize the reference to Red Shirts as being what turns out to be, if you do a fine analysis of the episodes, a bogus idea that if you're on an away mission on Star Trek and you got a red shirt, your probability of dying is higher. Okay? But that's sort of irrelevant because this, in these, I can't tell you too much of this or I'll give it away. In the space opera universe, there is a crew on a spaceship that has a very high incidence of death in the away team. So high in the away teams, so high, in fact, that some of the people on the crew become suspicious that there's something going on. Okay? And so they look into this, and indeed, something is going on. And I really shouldn't say anymore, or it's going to wreck it. I will say, do read the codas. These are almost like 3 little epilogues coming from different perspectives on the whole thing. I got partway through the first one, and I thought, man, is this really, he's really going on here. Should I really do this? The answer is yes, because the third one is awesome, and it wraps the whole thing up. So there you go. Red Shirts by John Scalzi.
Cool.
After your pick last year, I read Old Man's War. And you can read these very quickly.
Yeah.
That's actually next in my reading pile. I just finished Jonathan Strange and Mr. Norrell. Classic fantasy, but it's 800 pages long, and now I'm down for something.
Tell us if you like. I mean, Rich really liked Old Man's War. I thought it was okay. I thought it was a little corny at times, but let me know.
I think it was Scalzi's first novel.
I'm a sucker for space operas.
Me too.
You know, The Moat in God's Eye, okay?
Oh, that's very good. I like that. What's the author? What's his name?
Oh, I forget.
I like that very much.
What's that? Oh, Larry Niven.
Larry
Jerry
Niven.
Pornell.
There's a book, another one. I don't know if it's that one. They go to a planet where these like centaur-like creatures, right, with a human
Hyrule?
torso.
No.
What?
No, I made a silly joke that, okay, Brianne got it.
You know,
I got
because I
it.
said.
It's a Legend of Zelda has these
Anyway, so
centaur-like creatures.
the humans go and they're trying to interact, and it's difficult, and the whole book is about this. And then at the end, the centaurs, or whatever they're called, they start to interact with humans and learn their language. And they, at one point, share some pizza with them. And then later, at the very end of the book, one of the centaurs says, Pete, P-E-A-T-SA. He just phonetically pronounces it because he wants some pizza. I think that stuck with me. It's so funny. I think it's one of his kid books, actually.
Okay.
Which is really very good for everyone. All right, my book is by a Japanese author, very well-known, Yoko Ogawa. And I just stumbled on this because I really like Haruko Murakami, one of my favorite authors, and he hasn't written anything lately. I've read all of his stuff. So this came up as a recommendation. Mina's Matchbook. This is not sci-fi. This is not war. It's not violent. It is the most gentle and evocative book you will read. So let me read the summary because it's cool. In the spring of 1972, 12-year-old Tomoko leaves her mother in Tokyo and boards a train for Ashiya, a coastal town in Japan, to stay with her aunt's family. Tomoko's aunt is an enigma and an outlier in her working-class family, and her magnificent home and handsome foreign husband, the president of a soft drink company, are symbols of that status. The 17 rooms are filled with German-made furnishings. There are sprawling gardens and even an old zoo where the family's pygmy hippopotamus resides. The family is just as beguiling as their mansion. Tomoko's dignified and devoted aunt, her German great-aunt, and her dashing, charming uncle who confidently sits at the family's patriarch. At the center is Tomoko's cousin Mina, a precocious, asthmatic girl of 13 who draws Tomoko into an intoxicating world full of secret crushes and elaborate storytelling. It just makes the greatest atmosphere. It's unbelievable. Really calm. And for example, this pygmy hippopotamus. So her grandfather or uncle is wealthy. He's the president of this soft drink company. And he purchased this and he said, I paid for this hippo 10 Mercedes. It's very expensive. And it's an integral part of the book. Really, really good. I mean, just a gentle thing that you can read in a couple of days. And I like it. This is the first thing I've read from this author. So I'd like to try more. And, there's something about when Japanese is translated into English, it has a certain rhythm and gentleness to it. Even Haruki is very interesting in the English translation. I don't think there's anything like it in native English. So I really like that. But it's very atypical for me. I mean, there's no science. There's no any of that stuff. It's just a very gentle, and nothing really happens. Okay. Things happen. Their life goes on. She stays for a year and then she leaves, some things happen in the meantime, but it's very good. Anyway, we have two listener picks. Rona writes, Dear Vincent, in yesterday's office hours, you graced us with the poem by E.E. Cummings, Molly and Millie. My favorite singer, Natalie Merchant, put the poem to music. It was on her album and gorgeously illustrated book CD set in which she put many children's poems to music. My pick for you is to check out the Natalie Merchant song, which you can hear on YouTube, gives a link. Or better yet, get the Natalie Merchant book and CD combo. It's illustrated by equally awesome Barbara McClintock. Provides a link to her books as well. Always curious, Rona. So as I say at the end of each office hour, stay curious. And she does. Yeah, so lately I've been reading poems on office hours, and I've gone through a couple of my favorites. And yesterday, Wednesday, we did E.E. Cummings of the all lowercase, right? And run-on words and funny arrangements and stuff, but very cool. And Owen writes, for those who listen to Dr. Griffin's updates each week for advice on vaccines, the January 2026 issue of Consumer Reports has an article, How to Get the Vaccines You Need Now, that seems to do a good job of summarizing the information Dr. Griffin has been providing. After reading it, I would not be surprised if someone told me that some of the editorial staff at Consumer Reports are avid TWiV listeners. I think we have a lot of closet TWiV listeners for sure.
Yeah.
We have listeners all over the place.
All right, that is TWiV 1287. You can find the show notes at microbe.tv/TWiV. We can get your questions and comments and picks at TWiV@microbe.tv. And if you enjoy our programming, we'd love your support. Microbe.tv/contribute. Alan Dove is at alandove.com, turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
Brianne Barker is at Drew University, BioProf Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
Rich Condit is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
I'm Vincent Racaniello. You can find me at MicrobeTV. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.